For this lecture, we will go over the microscope and a little bit about the microscope and how to use it. At this point in your educational career, you're probably familiar with a microscope, and you probably understand that there are a variety of microscopes out there. And in the laboratory field, most of what you're going to use is going to be a compound light microscope or a bright field microscope. And so with these, you do have some magnifying lenses that, some different objective magnifying lenses, and of course you have your eyepiece oculars that do some magnification. And so we're going to learn a little bit about that total magnification when you combine those two lenses. And we're also going to talk about how the different resolution and focus in your microscope and different things that you do to your microscope kind of helps with that resolution and being able to visualize the different characteristics of the cell. So we're going to go a little bit more in depth of how to use a microscope than what you may have seen in your previous courses that you've done, if you've done any sort of science courses that use microscopes. And of course, depending on the courses that you took and the degree you took will depend on exactly what you learned about those microscopes. And of course, part of that resolution is going to be dependent on a couple of things. A numerical aperture is one of those things. And so this has to do with the amount of light that you allow through and how it hits that image or that cell on that slide, and then how that light is redirected, which will factor in of how crisp and clear your actual viewing field is in those cellular images on that microscope slide. And of course, it has a little bit to do with the distance that that slide is from that actual objective or that lens. So these things combined will determine the amount of resolution that you have on a slide. Now, to ensure that you know how to properly care for and use a microscope, you do need to understand the parts of a microscope. So the microscope itself consists of the main framework, which is going to consist of basically the hard structure bulk part of the microscope, which is going to include the base, the arm, and the stage. And then from there you have the illumination system, and that's going to be your light source, the things that control your light source like the condenser, the iris diaphragm, those things like that. When you control them, control how much light goes through, controls how much light hits the actual the slide itself and the cellular elements that you're going to see on the slide. So that's going to kind of factor into your resolution and how well you can visualize the various components of those cells. Then you have the magnification components, which is going to include your objectives and your eyepiece oculars. So these things combined will give you that magnification, and so you have different levels of magnification that we'll discuss. The focusing system, which is going to be your ability to fine focus or coarse focus in on objects. It's got to do with your depth that you are away from your oculars and making sure that you adjust those oculars to fit your eyes. So these are things that we will learn and go over when you're doing your campus lab. And you will use a microscope a lot throughout your studies in the medical laboratory field and throughout your career, so you should become very familiar with the microscope itself and the various components and parts of the microscope. You will be responsible for cleaning and caring for the microscopes, so you will need to learn how to properly use them and the various components of the microscope as far as what's acceptable for you to clean and what's acceptable for you to clean it with. So you do want to focus on learning the various pieces of the microscope and what those areas are called, because we will refer to these when we're working in the student lab. So when I'm going over, or your lab instructor is going over the various parts of the microscope or what you need to do some of the various things to care for that microscope and to set it up for your eyes, then we will refer to some of these various parts. So make sure that you are aware of what they are. Now, microscopes do not all look the same, but they do in general have about the same setup, and they do have the same components. So they might look a little bit different as far as where the levers are to control some of those things, but in general, they all do pretty much look similar to this. So you're going to have your objective lenses that all have a different power, your oculars, your diaphragms that control your light sources, and of course your focus adjustments. So all these are going to be pretty similar from one manufacturer to the next. And of course the microscope does use a light source. That light source is generally at the bottom base part of the microscope somewhere, and then that light source will go through a condenser. And then up through that condenser, it'll go through the substage lens system, which is going to be your lenses and things that are below where your actual stage is and where you place your slide. And then that light is going to pass through those condensers, and we're going to learn how to adjust some of those condensers to make sure that you get your optimal light path coming through that specimen. And then of course above your specimen you have your objective lenses, and your light source is going to pass through those. And that's going to allow that light to reflect through and around whatever cellular component, or in the case of like a urine sample, it could be a crystal or something like that. And so as it passes around or through that actual slide, then it will come up through your objective lenses, and then it'll come through your ocular lenses, which also has a magnification in it. So you're getting magnification on top of magnification, and then of course you can visualize it with your eyes. And again, most of what we're going to use in the lab setting will be a bright field microscope. If you're doing some more detailed work or some various tests that require other microscopes, then you may occasionally use a different type of microscope, but primarily this is what we're going to use, is a bright field microscope. And so to make sure that you get the most benefit out of your microscope, then there are things that we need to make sure that we know how to do, which is to properly care for them, keep them clean, and to make sure that we set it up properly to meet our needs for our specific eyes. So these are all things that we're going to learn throughout this particular module. So caring for the microscope includes understanding how to properly clean the microscope. So you want to make sure that at the end of the day, when you put your microscope up, that you have made sure that you've cleaned up any oil residual or anything that may be on it. If you're getting the microscope out for the first time, we're going to learn how to clean them off, because sometimes when they've sat for a little bit, even though we have covers and things to go over them, they do get a little dust on them. So we want to make sure that we clean them properly, remove all that dust, make sure that we don't have anything that's blocking the movement of our stage or anything like that, so any kind of dried up old oil or anything. And so by making sure that we keep the microscope clean, make sure that we don't have any scratches or anything on our lenses, this is going to help us to view our slides so that we don't have anything kind of interfering with our field of view and magnification. Now, once we ensure that we have a microscope that is clean, then we're going to make sure that we properly align it. So we're going to make sure that all of our light sources are lined up properly, to make sure that we get the optimal light source coming through our specimen slide. And then of course we're going to learn how to do the proper adjustments. There is light intensity based off of the specific objective lens that you use. We got to make sure that we have everything lined up properly, that we have our stage at the proper height to make sure that we get the proper amount of light coming through our specimen. And of course we're going to learn how to do the proper focusing. So we're going to learn how to first do a coarse adjustment, and then once we get our specimen in our field of view, then we're going to do a fine adjustment, and we'll also do some adjustments to our light source. Now, some of the other different microscopes that you may encounter while you're working in the lab may be a dark field microscope. And so what this will do is it will use a scattered light pattern when you're looking at these objects on the microscope. And then what it's going to do is create a dark background, and then you're looking for the reflection of light on those objects in order to illuminate that particular cell or object that's in that microscopic field. You have differential interference contrast microscopes, which gives them sort of a 3D type appearance. You have electron microscopes. This provides a really more detailed magnification and resolution. And then within that you have a couple of different types of electron microscopes with a transmission and a scanning electron microscope. And so you're going to see these in more of like your larger research type facilities where you're going to see these electron type microscopes. There are fluorescent microscopes, and so what these do is they're looking for anything that may have been fluorescently labeled. So these will be usually like an antibody type fluorescent label in which you're trying to fix something to a slide and then try to tag it with a fluorescent label to see if it's present, and then you're looking for a fluorescent glow when you're actually looking at that slide. Phase contrast microscopes, these use an increased contrast on an unstained specimen. So these may be used when they're trying to look at specific casts and things in urine. And with these microscopes, the tech that's using these microscopes needs to be very proficient in their techniques in order to make sure that they understand how to go from a bright field to a phase contrast and what exactly they're looking for in those fields when they're using that particular microscope. There are also polarized microscopes. Now, polarized microscopes are actually kind of fun. We use these to look at more specifically crystals and body fluids. And so when you look at the crystals and the body fluids, depending on how that light path passes through that crystal and the color that that crystal illuminates based on those light fields and the direction that that crystal is laying, then it kind of helps you identify the particular crystal. So we use these when reporting this back to the physician, because based on the crystal type that may be in that fluid will help them to determine exactly how they need to treat that patient, or what particular crystals tend to be the type of crystal that's present in that patient could alter the diet or various other things in the treatment plans. So that's why these particular microscopes are used. We're starting to see a lot more of the digital microscopy in the medical laboratory field. And what this does is it's actually, you'll have some analyzers that have this component tied to the analyzer. And so the analyzer will make a slide, and it will go ahead and look at the cells and pre-classify what category to put those cells in. Or a urinalysis analyzer will kind of do the same thing. It'll do a slide out of the urine and it'll run it through digital microscopy unit where it will actually start classifying crystals and cells and various things in it. And then from there, the tech will actually review those images to make sure that it looks like that it was placed in the right category. I've never used one of these, but I have heard from other students, and I have seen them in some clinical sites. And kind of the consensus with these is it's great. It kind of saves time, but it also causes a little bit more work for the tech because it doesn't always classify the cells properly. But this is something that's kind of a newer technology, and you probably will at some point in your career start seeing this a little bit more. And so with the digital microscopy, it does allow the storage of those actual images. So whereas now we kind of keep the microscope slides once they're made, we save those for a couple of years before they're discarded. This actually stores digitally stores those images so that they can be reviewed at a later time. And you know, now with things being more electronic, then that database can be preserved for quite some time.